In this video, I will discuss artificial method of vegetative reproduction. There are different methods like cutting, grafting, budding, and layering. Let's talk about cutting first. It is a portion of the root, stem, or the leaf, common method for artificial vegetative propagation, and it is found in sugarcane, roses, citrus, and grapes. Procedure: a cutting is put into the soil. Nodes found lower in parental body morphologically are put into the soil, and higher nodes are kept high. Adventitious roots are taken out at the lower nodes. Special growth hormones like IA, IBA, and NA are applied in minimal concentration or in dilute concentration. Favorable time is spring. So what happens is, like, let me explain cutting. Cutting are the portion of the root, stem, or the leaf from the parent. So this root, stem, or leaf, farmers, what they do, they cut and place them in the favorable condition or in the soil, and these cut portions of the parent, which is the root, stem, or leaf, develops into a new plant artificially. This we find in plantation of roses, sugarcane, and grapes artificially. This will give new variant of plant, and it is the very fastest method of germinating new plants artificially. Now let me talk about the procedure. This cutting is actually taken from a parent plant. Let's say this is the parent plant. And from this parent plant, this cutting has been taken, and this cutting is now put into the soil. So the nodes, these are the nodes, that is a junction, which are found in the parent plant lower morphologically are put in the soil, while the higher nodes are kept high. And in order to firmly attach to the ground or the soil, these cutting will develop adventitious roots to attach to the soil, but these roots are taken out or taken off from this cutting portion of the parent plant. Why? Because if these adventitious roots are not taken out, then this will prevent the germination of the new buds. So once these adventitious roots are taken out at the lower nodes, and special growth hormones like IA, which stands for indole acetic acid, IBA, indole butyric acid, and NA, naphthalene acetic acid, are added in dilute concentration, these buds will develop at this cut portion of these nodes, and these green color small buds will develop into a new plant. So this is called as cutting. Now grafting. It is actually a procedure that is followed between two plants of closely related varieties. This we have seen in germinating desired variety of roses. Different colors of roses, The stalk, that is the root system, desi mango, and the scion, shoot system, desired variety of flowers or fruits, the cherry mango. So here you see I have mentioned with two colors. This is the stalk portion of one variant, and this is the scion, that is the another portion of another variant. So these two parts, stalk and the scion, are joined here, and some hormones are applied to produce this stalk and the scion. Once they are joined by some wrappings or nailing and applied with some hormones, this joint this fused form of stalk and scion will germinate into a desired variety of plant species. So the root system from suppose a plant, suppose a tree which is having a strong root system. So that root system is taken out from one plant, and another variety of plant which is having good it shows good flowers, or it produces large quantity of fruits. So these two things are joined together so that the new plant which is formed is having strong root system and strong shoot system, and this new variant is formed only by the process called as grafting. Here the union of the scion and the stalk, or the stalk and the scion, results in organic connection between them after some time, and the cut surfaces are held tightly as I mentioned by wrapping, nailing or some other method. All shoots arising from the stalk should be removed. So the root system is known as the stalk. So any shoots that will arise here is not required because this is the root system. This has to be strong. So these unwanted shoots are taken out because we want a new plant from the scion, not from the stalk. So this is called as grafting. Now about budding. A bud with small portion of bark is taken from the stem of a plant and then inserted into the small slit in the bark of stalk. Both are tight. Buds of stalk are not permitted to sprout. Both are tight. Buds of the stalk are not permitted to sprout. Example, rose and plums. In budding, what happens? You see, this is one variant of plant. So here green colors are the buds. Okay, so these buds along with some portion of the bark is a covering of the plant. So this bud, green color is the bud, with a small portion of the bark is taken from the stem of a plant, and this is inserted in a small slit in the bark of stalk of another plant. You see, slit means a small cutting, because to adjust or fit this bud with the bark in the new stalk, so slit has been made. So in this slit this is attached, okay, and thereby it forms a new variant, and both are tied obviously. So this happens in rose, plums. Now layering. The stem branch used for propagation layer remains attached to parent till it has produced adventitious roots. It is of two types, ground layering and air layering. Now the stem branch for propagation layers remain attached to the parent till it has produced adventitious buds. You see here the stem branch which is used for propagation remains attached to the parent. This is parent as whole, and this is the newly formed branch. It is attached to the parent till it has produced adventitious roots. Once this has produced adventitious roots, it is now separated from the parent to develop into new plant. So it is of two types, ground layering and air layering. Now talking about ground layering, it is the stem is selected to bend down to the soil. Portion touching the soil is cleared of the leaves. An injury is made on it by cutting a ring around or by beginning an oblique cut on bent portion of the layer at the node, or by giving a V-cut at the lower node of the bent portion of a layer. Auxin hormone is applied and adventitious roots are formed thereby. So it is found in jasmine, strawberry and some other variants of plants. So let me discuss about this thing. So this is the ground. Use different colors. This is the ground or the soil, and this is the plant. This is the plant. So in step 2, what happens? This portion is selected and bent down to the soil. So this is selected and it is bent into the soil. In the third stage, an injury or a cut is made onto the bent portion of the stem, or an oblique cut is made artificially, or a V cut at the node. And hormones are, and most importantly, here you see leaves are there, right? So the leaves, because this portion is used for bending, so the leaves are taken out or cleared from the portion that is touched in the soil, because if this leaf is not taken out, then it will hamper in the formation of, or hinder in the formation of, these adventitious roots. So once this is bent and leaves are taken out and injury is made and hormones like auxins are added, so after some days or after some days adventitious buds will form, and this again will form a new plant once it gets detached from the parental plant. This we have seen in jasmine and strawberry. This is very important. Ground layering. Now comes the air layering. Air layering. Bark of the chosen branch is cut by girdling or ringing near the bark. This initiates root formation. Treated with hormone auxin. A ball of sphagnum moss is placed around the cut portion, kept moist and enclosed in a polythene bag. I have already mentioned these are all artificial methods of propagation. When the root appears, the stem is cut below the leaf level of the roots and plated. This method is also called as lychee. We have found this air layering in lychee, citrus, pomegranate and guava. So here in air layering, bark of the desired branch is cut, and this cut portion of the branch, which is the bark, will initiate root formation, and after these are treated with hormone auxin. To keep this thing moist, a ball of sphagnum moss is laced or tied around the cut portion to keep it moist and are placed in a polythene bag. When the root appears from this bark,